here. In the name of Allah, the most beneficent and most merciful, I hope you are not tired. So it's the last session and last but not least. And I want that to wake you up with giving an appreciation for the long wait and patience. So clap for yourself and wake up. Thank you. So I will be focusing more on the work related to this project we are going to do in Islamabad, because as one of the site where we are doing experiments on vegetable storing on wastewater, and that is also backed by some studies and surveys that was conducted in 8 different districts of Punjab. And that detail will be presented in Islamabad. We have arranged another seminar over there where we can present that study. So we will be focusing in this presentation on the typical experiments that we are conducting. So in the context of gastroenteritis, we want to study that how the pathogens, heavy metals and other contaminants from sewage sources, they are going into the food chain. So this slide we have seen in almost all the presentations. So we are focusing mainly in this with the agricultural component of that, okay, in this chain. And the objective of our study is to understand the efficacy and how the alternate, there could be some like waste treatment, what waste alternate options are possible, where the water is scarce and only sewage wastewater is the option for the farmers to do it, especially in the peri-urban areas of industrial cities like Islamabad, which is the 3rd largest populated city of Pakistan. And we will see in detail about the pathogens, and we have brought the samples of different vegetables for testing the pathogens here, and we will continue the work, so this will make a chain through this that we will complete to understand the whole studies that how vegetables are being affected and how they come into human contact through the use of vegetables. So various activities of the site selection, I will show you some pictures and videos in this. So this is the site which is in Islamabad. This all wastewater collected from the hospitals, from the different departments, that is being collected at one place, and from there it is pumped to the drainage. So we'll be using that how much contaminated is the university's water and how much is the contribution of university wastewater toward the drains. One site, this where I was showing you a picture. This is one place. This is the trial location 1, which is in Islamabad, with this latitude and longitude. The second site, that is Chitera Farm, and here you can see there are some big ponds. This is the sewage treatment plants, and these are very big ponds. They are spread over several kilometers, and all of the wastewater from industries and different household water, and that is in big channels that is coming towards this place. So from this treatment plant, then this water travels. If you can track with the Chitera Farm and these ponds, if you track through the Google Earth, you can find that finally this wastewater is ultimately going into the river Chenab. This is the third site, which is in Lakhiamwala village, because we have different sources, because Islamabad, as I mentioned, is a big city. On the one hand side, there is a big drain, and on the other side of the city, there is another drain. The earlier shown enters finally into river Chenab, and from the wastewater from this area that enters into the river Ravi. So this wastewater ultimately travels and go into the rivers. So the plantations we did, we chosen 3 different vegetables, and among them one is tomato, and we prepared the nursery, we prepared the media, and then we grown the nursery plants, then we transplanted them into the field. Here is the team that is preparing the things. This is a germination of tomato. You can see some plants, they are growing, and then after a certain time, when they are ready to be transplanted into the field, then we transport it there. And here is the land preparation methods that we chosen the bed planting methods. And here you can see in some parts we prepare that manually because of pathway. And here this you can see is the source of sewage water. Inside from the labor is working here to set and make partition different segments. And one segment was only irrigated with canal water, one segment was only irrigated with the sewage water, and one segment was treated wastewater with biochar. So there are three different type of options we have. Firstly, this was also prepared with the tractor. So the bed making process, the tractors are working to prepare the field for. And if you come to festival, you can enjoy the ride on the tractors as well. So this is a wastewater irrigation. You can see the color of the water is very dark color than normal irrigation water. Here you can see it's traveling from the channels and going all together into the field. So this is all the layout where we grown vegetables. Okay, and then the seed was sown, especially for the lettuce and beetroot. Seed was sown directly, and in case of tomato, we prepared nursery plant first, and then we transplanted. And here you can see the transplanted tomato seedlings that we transferred from the trays. And here you can see the output. This is a field trial, and here you can see lush green field of lettuce. It was great growth from the sewage water, contains a lot of nutrients, a lot of pathogens also. So nutrients we are testing over there, and for pathogens we have brought the sample to be tested by Dr. Imran here. And beetroots, we have brought the samples of beetroots, and also the tomatoes, we have brought the samples here. So the idea behind choosing only these is, the major thing is that these three vegetables are consumed uncooked. These are consumed as fresh salad. So they have chances of, more chances to go into the food chain and for direct human consumption. And for lettuce, we use the leaves. So we will try to understand the uptake mechanism as well, how the plant's physiology functions for the uptake of different kind of nutrients and heavy metals. So how the leafy vegetables perform, how the root crops perform, how the fruit crop perform. So we'll be comparing these three types of vegetables, that how they are performing and how they are taking up pathogens or nutrients. Then there was biochar application into the field. So you see here, firstly it was spread, and then it was raised up manually, and so the roots, the water goes through the roots by passing through the biochars. There were various data and parameters we recorded. A lot of these includes growth and yield parameters, which shows about the height of the plant, leaf length, root shoot length, and mainly we focus on the root study, that what changes occur in the roots, how they are spread, and how they are uptaking the pathogens, what are the sources of interaction. We will be also doing some microscopic studies of the roots. Then we will be testing physiological and biochemical analysis of the plants, chlorophyll contents, carotenoid, electrolytes, leakage, and various kind of antioxidants, anti-stress elements. And we will be doing approximate analysis. Some of the results I have prepared and I will show you here. This is here the fresh weight in lettuce in case of control, that is canal water. You might see that the fresh weight where biochar was applied, fresh weight was comparatively more. The dry weight in lettuce was also more with the biochar treatment. And for the tuber root, tuber beet, the beetroot, the biochar was also performing good. With the number of fruits in tomatoes, there were more number of tomatoes. You can see here 20 tomatoes, here 28. So it's a big difference with the application of biochar. So biochar is ameliorating with the soil and sewage water. Then photosynthetic pigments in tomatoes, the total chlorophyll content was more with the biochar. Total free amino acids was also more in the same sequence, in the same trends. Total sugars were comparatively more. Then proline content, because this is produced in stress condition, so this is less in biochar. When biochar was there, then plant stress was delayed. And likewise, osmolytes in tomato and MDA, malaldehyde content, that was also reduced. That was also representative of the stress reduction. Then electrolyte leakage, that was also less in the biochar. That shows these cell membranes permeability. Here was the soil analysis, and various kind of analysis were performed. And after the complete harvest of the experiments, then we will analyze again to compare what was the impact of sewage treatment compared to the biochar and for all these parameters. And then here is the invitation for 25th. We are organizing a seminar on biochar, so you are all welcome, and also you are welcome to have the tractor ride. Any question?
Thank you, Parth, for sharing a very detailed and related presentation with the project. First question, the water that you mentioned, wastewater, is it treated water or an untreated water that you are using for irrigation purpose?
No, we are using wastewater, directly wastewater.
Direct raw wastewater?
Yes.
Okay, raw wastewater.
And also the biochar one. We are using both options. Treated with biochar.
Treated with biochar.
Yeah.
Okay, you are using raw wastewater.
Yeah, so wherever I mentioned biochar, that is the wastewater is treated with biochar.
Okay, what is the probability of the capillary movement of the water within the plants, and can we rule out the movement of the microorganisms within the root, shoot and the leafy part of the plants?
Yes, there are chances of contamination as well. And we are trying to find out firstly that how the plants are behaving, what are the roots level interaction, and we will be testing for the pathogens by doing the culturing process that earlier we have discussed in the meeting. So we will be doing part of the culturing and to see how much load of pathogen is there. We'll see the different treatments with the direct wastewater and with the treated wastewater, how much is the bacterial load present in those plants.
Again, what is the back origin of this raw wastewater? It's from the university residential, or what is the nature of this raw wastewater?
Yeah, we are using 3 different sources. We have 1 trial at the university's field and 2 trials at the farmer's field. So at the farmer field, there the industrial and household water is mixed. So industrial water is also part of that. So the big ponds I have shown, that is all the water from the reservoirs, almost half of the portion, all the industrial water that goes into that drain. So we are basically studying 2 major drains. One is Pahadan drain and one is Madhuana drain. So those 2 drains are the major focus. Those are 2 big drains which collect almost half of the water on the right side of the city and half of the water on the left side. And both of these drains, one goes into the Chenab River, one goes into the Ravi River.
Do you have any picture related to the growth of the plants with regard to these 2 different nature of the wastewater currently?
It's not shown here, but the data is here, but the pictures are not here.
Okay,
I can show you the picture
later. Thank you.
It's in the Dropbox. You
Okay,
can see
because I just want to see the comparison, the growth rate with regard to the industrial wastewater growth plants and the university residential area wastewater plant, because we assume that there is higher toxicity in the industrial wastewater. So what is the competition for the plant to grow in comparison to the other one?
Yes, sir. The whole growth data will be available. So still the crop is in the field. We have partially harvested that, but still there is some because for the field visit, we can show you over there during the visit to Pessoa.
Last question, I see the tomatoes. So what about the pulpy and the juicy part? Because of the capillary movement, is there any possibility the accumulation of the heavy metals, the antibiotics or other toxic materials or even the microorganism to be within the leafy and the juicy part of the tomato?
Yeah, we have preserved the samples, we have not tested yet. We have preserved the samples, we are in the process of doing so the data available I have shown, and rest of the data we are preparing. And the samples are also here, so some of the part will be done at next.
Thanks very much for that part. It was fascinating to see the results of the experiment. We're really excited. But yeah, no, so I thought there were a couple of interesting things. One was you had some measures of stress in the crop, and it looked like those might be higher with the wastewater. So in comparison to the control. So is it possible that the wastewater is actually stressing the plants?
Yes, it is stressing. In some of the parameters, they are stressing. And with the intervention of biochar, the stress level is less.
Okay. And then my second question is a sort of slightly technical one for you and Ruben. So you've stored the samples such that we could test for heavy metals, right?
Yeah, we are processing the samples. We have given the samples. They are under testing. We have given the samples for testing.
So where are they being tested?
In the university, we have the facility for testing heavy metals.
Because it would be really good to
Yeah, we'll be showing you that high-tech lab as well during your visit to Pessoa.
Yeah, because what would be nice, I suppose, might be to do the microbial resistance of the same samples.
Yeah.
As the ones you're doing for heavy metals. So we should discuss that.
Yeah, I have recruited one PhD student especially to do that work related to heavy metals and these parameters. These two ladies from Pessoa, they are here, and one of them is being placed on the relevant subject. She can also answer some of the questions.
Yes, can you introduce yourself, please? Right there.
I am Vikram from University of Agriculture, Islamabad. I am under the supervision of Dr. Fahad Rasool. I am doing PhD in what?
She has prepared her synopsis. We will share with you that what we are planning, so you can propose some more things in your study plan. Yeah, that would be good, so we can add some more part of that.
Thank you very much for the presentation. I just had a question actually when you said like about the sample that you got here. When you say the sample, do you mean like we only have the leaves, the fruit, or some soil, or the rhizosphere? Which sample did you, what did you sample actually from each crop?
We have brought leaves of lettuce, roots of beetroots, the developed roots, and we have brought some tomatoes.
Okay.
Yeah. Of all the treatments, yeah, and from all the treatments, we have the tomatoes from directly irrigated with wastewater, with biochar intervention, and with canal water. We have all the samples. So afternoon, we will hand over you.
Thank you, Doctor, for a nice presentation. You have invited all the participants to have a checker ride, but you haven't invited them for tasting the tomatoes and eating all those stuff that is actually you are preparing.
Yeah, we have food arrangement, and you will enjoy all the recipes over there. We have many more options apart from these vegetables.
No, it would be interesting to see whether consuming this lettuce one can survive or one can. It would be a kind of bioassay.
If you can volunteer to be an experimental organism, then
No, I think team members must take the lead actually. Volunteers first because they are part of the team actually.
yeah, maybe after the completion of project.
Okay, just one short question actually. I wanted to know whether the treated and untreated wastewater that was characterized with respect to the presence of heavy metals, antibiotics, etc. So do you have detailed analysis just to know what was the composition or even with respect to the organic content or even COD in terms of COD or BOD actually?
Yeah, it's a good question. We are doing the profiling of all the sources of water, and we are also doing the fraud experiments on site by you see on the micro scale as well, and also the field studies. Comparative analysis of both will be happening, and the profiling will be shared in the next workshop.
And hopefully soil characterization as well with respect to
This
the
all is being processed. because-
organic matter content. As early in our earlier presentation we have seen that that's also a critical component with respect to the mobility of the
Most of
environment.
our samples are in process. Those which were ready, we have presented. Many of the samples are in process, and still the crop is in the field. And we have recruited one PhD dedicatedly for this work. So we'll be having many more site studies along with field studies. We'll be having port studies as well.
Thank you.
Can I ask a quick question, please?
Yes.
Yeah, sorry, Fadia. I've been trying to listen carefully, but I did not see your slides. But a question about stress that you mentioned in the crops. Did you also measure the salinity of the water?
Yes, we are measuring the EC and pH both. Yeah, we are measuring that.
Okay. And did
In
you
the soil,
see?
in the water, and also in the plant.
Okay. And when you compared the different sources of water, did you see a greater difference in the industrial one compared to the less industrial or domestic ones that because you've got 3 sources of rainwater, right? You said.
Yes, we have 3 different sources of water, and we'll be presenting as soon the data is compiled, that will be shared, and
okay.
we'll be able to. Yet all the thing is not analyzed because we have done this for the first season. We'll be doing that replicated trials we have planned for 3 to 4 different seasons consecutively to
Okay.
see the variations.
Okay. Thank you.
Any more questions?
Gratitude, Dr. Fahad Rasool, for enlightening us and sharing your experience. I hope that our audience got much educated beyond their own understanding related to impacts of sewage on crops. Now, this brings us to closure to day 1. I think we have covered almost everything on the list, and I'm pretty much sure that the knowledge and research directions shared by our delegates and knowledge ambassadors were of utmost significance to all of us over here.